Welcome to the deep dive. Today, we're taking a plunge into something truly incredible: how animal bodies operate, specifically their amazing internal communication networks, the endocrine and neuroendocrine systems.
Yeah, it's fascinating stuff.
And to kick us off, let's look at one of nature's most extreme masters of physiological adaptation, the grizzly bear.
Oh, absolutely. It's an astonishing story of survival. Imagine these powerful animals prepare for up to, what, 7 months of hibernation?
7 months, yeah.
And they do it by first becoming what scientists actually term reversibly obese. They just gorge themselves in the fall, meticulously storing fat to draw on during those long, lean winter months.
So they're storing all this fat, but how do they control that storage and then, release it so precisely? What's the master switch here?
Well, it really comes down to insulin and how their bodies masterfully manage its sensitivity. It's quite clever.
Insulin sensitivity.
In late fall, right before hibernation, their fat cells become, like, exquisitely sensitive to insulin. Yeah. Super
So
responsive.
they just soak up everything.
Pretty much. It allows them to eagerly take up and store every nutrient as fat.
Mm.
But then, as they enter their dens, those very same fat cells become dramatically insulin resistant.
Whoa, okay. So even with insulin still floating around, the fat cells basically just ignore it,
That's the gist of it, yeah.
which means the stored fat can be broken down for energy, fueling them through hibernation. Clever.
Exactly. And then, come spring, when they emerge, those cells gradually become insulin sensitive again. The whole cycle reverses.
So the core insight here is that insulin resistance isn't always a disease state. For the grizzly, it's actually a finely tuned survival mechanism.
Precisely. And understanding how they flip that switch, well, that could be a game changer for treating type 2 diabetes in humans, where people are often obese and, crucially, insulin resistant.
Right. It potentially offers a completely new therapeutic angle, maybe.
It really does. It's a perfect example of why looking at these sort of extreme animals is so valuable.
Yeah, that truly highlights the potential. So for this deep dive, we're drawing insights directly from a key chapter of Animal Physiology, the fourth edition by Hill, Wise, and Anderson.
Great resource.
Our mission today is to unpack the fundamental concepts, the mechanisms, the systems that govern how animal bodies work. You'll get, hopefully, a clear, engaging understanding of how hormones orchestrate life.
From a bear's deep slumber to, say, an insect's dramatic metamorphosis.
Exactly. We'll be emphasizing how different species adapt, the why behind these adaptations, and importantly, how we learn about them through sometimes really clever experiments.
Yeah, the experimental side is often just as fascinating.
Okay, so let's dive into the fundamental alphabet of communication that makes all this possible. Chemical signals. How do cells in a body actually talk to each other, near and far?
Well, there's a whole spectrum of ways, really. On the shortest range, you have what are called autocrine and paracrine signals.
Autocrine and paracrine. Okay.
These diffuse only locally. Autocrines act on the same cell that secretes them, while paracrines influence neighboring cells. They don't enter the bloodstream.
Gotcha. Like local memos? What are some examples?
Think of things like cytokines, which can direct cell development or immune responses. Neuromodulators fit here, too. Some cytokines also play a role in angiogenesis.
Angiogenesis. That's new blood vessel formation.
That's the one, vital for growth and wound healing. So these are really important local regulators.
And then, of course, there are neurotransmitters. Most people have heard of those.
Right. The rapid pinpoint messengers we know from the nervous system, they cross a tiny synaptic gap for quick effects and are usually inactivated just as quickly. Very specific, very fast.
And finally, the stars of our deep dive today: hormones and neurohormones.
These are the endocrine chemical signals. They travel long distances in the blood, circulating throughout the entire body.
Which means they can affect lots of cells.
Exactly. Because they're in the bloodstream, they can influence large populations of target cells as long as those cells express the specific receptor molecules for that hormone. That's the key.
So if I'm understanding this, a hormone is a chemical substance. Could be from a non-neural endocrine cell or even a neuron. It's carried by the blood, and it works at really low concentrations on distant cells.
Perfect summary.
And neurohormones are just hormones released specifically by neurons.
That's it.
So that broad reach but kind of delayed response is a key distinction from those faster, localized neurotransmitters.
Exactly so. And the cleverness of how that widespread effect is controlled lies in the concept of target cells that possess specific receptor molecules.
Kind of lock and key.
Sort of, yeah. Think of it like a highly specific molecular handshake, or maybe a perfect puzzle piece fitting into another. Only the right hormone key can interact with its unique receptor lock on the target cell. to trigger a response.
A great example you mentioned is thyroid hormones. They have such widespread effects on metabolism, structure, development.
Because so many different cells throughout the body possess receptor molecules for them. They have lots of locks.
But the sensitivity of a target cell isn't fixed, right? It can change.
Precisely. A cell's sensitivity to a hormone can shift through upregulation, where it actually increases its number of receptors.
Makes itself more sensitive.
Right. Or downregulation, where it decreases them, becoming less sensitive. And importantly, many cells can respond to multiple hormones if they express different types of receptors for each.
So it's really versatile. Allows for incredibly nuanced regulation depending on what the body needs at that moment.
You got it.
Beyond receptors, the actual amount of hormone in the blood also matters. How is that balance maintained?
Yeah, the concentration. It's a continuous balancing act between how fast the hormone is secreted into the blood and how fast it's removed or broken down.
Removed by?
Usually by degradation, typically by organs like the liver and kidneys. This removal rate is indicated by a hormone's half-life, the time it takes for its concentration to reduce by half.
And that varies a lot.
Oh yeah. Steroid hormones often have half-lives of hours or even days, while some peptide hormones might only last minutes. Big difference in how long the signal persists.
And I remember reading that some hormones even get a bit of an upgrade after they're secreted.
Yes, that's called peripheral activation. A good example is thyroid hormone again. It's secreted mostly as T4, but many target cells then use enzymes to convert it into T3.
And T3 is the more active form.
Right. It's like sending out a precursor that gets activated right where it's needed.
Yeah.
Very efficient.
Okay, so if we group them by their chemical nature, hormones fall into three main classes. If I had to remember one thing about why these classes matter differently, what would it be?
Good question. I'd say it's all about how they get into the cell and how fast they work. Steroids are kind of slow but powerful. Peptides are generally fast but need surface access. That really dictates their whole physiological role.
Okay, slow versus fast, inside versus outside. Got it. Let's start with the steroid hormones.
Okay. Steroids are all derived from cholesterol. Think of insect molting hormones like ecdysone or vertebrates, sex hormones, and glucocorticoids like cortisol.
And because they're made from cholesterol, they're lipid-soluble, fatty?
Exactly, which means they can pass right through cell membranes. They don't need a receptor on the outside. They bind with receptors inside the cell, either in the cytoplasm or the nucleus.
And once they bind to those intracellular receptors, they form this hormone receptor complex, which then does what?
It essentially acts as a transcription factor. It goes to the DNA and alters gene expression.
Meaning it tells the cell to make new proteins.
You got it. And that takes time. So this leads to a delayed but often very long-lasting response, maybe minutes to hours or even longer.
But there's a nuance, right? Some steroids can act faster.
Ah, yes, good point. While most steroids work this genomic way, altering genes, some, like aldosterone or estrogen, can also bind to cell surface receptors for more rapid non-genomic effects, sort of behaving more like the water-soluble hormones in those cases.
Interesting. And how do they travel in the blood if they're fatty?
They're typically transported bound to carrier proteins, keeps them dissolved and protected. And importantly, they're generally synthesized on demand from precursors, like cholesterol stored in lipid droplets. They aren't usually stored in vesicles like other hormones, and they just diffuse out of the cell when made.
Okay. Next up, peptide and protein hormones. Big category.
Huge category. These are chains of amino acids, varying greatly in size. Examples include insulin, growth hormone, antidiuretic hormones, lots of familiar ones.
Is water-soluble the opposite of steroids?
Correct. So they can't just slip through the cell membrane.
Yeah.
They act by binding to cell surface receptors.
And that binding then triggers stuff inside.
Exactly. It regulates ion channels or activates second messenger systems within the cell. This primarily changes the activities of proteins that are already there.
So not making new proteins like steroids, but modifying existing ones. That sounds faster.
Much faster, generally. Responses are usually within minutes.
And their synthesis is quite a detailed process, isn't it? Not just made on demand.
Indeed, it's more complex. They're synthesized at ribosomes as large, inactive preco-hormones. Then they get processed through the Golgi apparatus, stored in vesicles, and finally secreted by exocytosis, which is often calcium-dependent.
Insulin is a prime example, right?
Perfect example. It starts as a large, inactive molecule, proinsulin, that gets precisely cut down to its active A and B chains. Then it's stored and released.
And there's that C-peptide part that gets cut off.
Right. And interestingly, that C-peptide is released right alongside active insulin 1-to-1. Clinically, doctors sometimes measure C-peptide levels because it hangs around in the blood longer than insulin itself. It gives a clearer picture of the body's own insulin production, separate from any injected insulin.
Clever diagnostic tool. Okay, and finally, the amine hormones.
The third class. These are modified amino acids. Melatonin, for instance, comes from tryptophan.
Sleep hormone.
Yep. While catecholamines, like epinephrine, norepinephrine, dopamine, those are the fight-or-flight ones, and also thyroid hormones, are derived from tyrosine.
And their properties are kind of mixed.
They are. Their solubility varies. Melatonin and catecholamines are water-soluble, but thyroid hormones are lipid-soluble, like steroids. So they can bind to both surface receptors and nuclear receptors. And often they play dual roles. They can be neurotransmitters in the nervous system and hormones in the endocrine system.
Very versatile molecules. Okay, so we've learned the language of the body with these different hormone types. Now, who's in charge of orchestrating these messages? Let's move to one of the body's most crucial command centers, the vertebrate pituitary gland.
Right, the pituitary. Sits just below the hypothalamus at the base of the brain. It consists of two really distinct parts, each with a unique connection to the brain.
Let's start with the posterior pituitary, the neurohypophysis. That sounds nerve-related.
It is. It's essentially a direct extension of the hypothalamus itself. It's neural tissue.
So specialized large neurons, called magnocellular neurons, actually have their cell bodies up in the hypothalamus.
That's right.
They synthesize hormones like vasopressin, also known as ADH, antidiuretic hormone, and oxytocin.
Yep, those are the two main ones.
And these hormones then travel all the way down the axons of those neurons.
To the posterior pituitary, which acts as a kind of storage and release site. It's called a neurohemal organ.
And they're released directly into the bloodstream via exocytosis when the neuron fires.
Exactly. Vasopressin, for instance, is crucial for water retention, responding to signals about blood volume or osmotic concentration. Oxytocin plays well-known roles in uterine contractions during birth and milk ejection during suckling.
And the structure of those two hormones is fascinatingly similar, isn't it?
It really is. They only differ at 2 amino acid sites in most mammals. Tiny difference. Yet these subtle changes lead to profoundly different and vital functions. It strongly indicates they share ancestral origins, and it's a prime example of evolutionary fine-tuning.
Amazing. Okay, now in contrast, the anterior pituitary, the adrenal hypophysis, totally different.
Completely different. It's non-neural endocrine tissue, glandular tissue.
Made up of different cell populations, each secreting specific hormones, peptides, proteins, glycoproteins.
Right, and these hormones are broadly categorized. You have direct-acting ones like growth hormone or prolactin, which affect various body tissues directly.
And then the tropic hormones.
Yes, tropic hormones control other endocrine glands. Think PSH, thyroid-stimulating hormone, ACTH, which acts on the adrenal cortex, LH and FSH, the gonadotropins acting on the gonads.
And these tropic hormones are critical not just for making the target glands secrete their own hormones,
but also for maintaining the actual health and size, the vigor of those target glands. If you deprive a gland of its tropic hormone, it'll actually shrink, it'll atrophy.
Wow, so how is the anterior pituitary controlled? It's not directly connected by nerves like the posterior part?
It's controlled by the hypothalamus, but indirectly. Through smaller neurons, parvocellular neurons, in the hypothalamus that secrete releasing hormones, RHs, and inhibiting hormones, IHs.
Okay, releasing and inhibiting hormones. And how do they get to the anterior pituitary?
They get there via a specialized, dedicated vascular pathway, a little network of blood vessels called the hypothalamo-hypophyseal portal system.
A portal system, like a direct highway.
Exactly. It quickly transports these RHs and IHs directly from the hypothalamus down to the anterior pituitary. This system is crucial because it ensures these vital signals reach their target without being deleted in the general circulation, allows for rapid and precise action.
So if the brain is our central nervous system and the endocrine system manages widespread physiological changes, how does this portal system act as a crucial interface between the two? It sounds really important.
It is the crucial interface for this part of the system. It's the brain's direct line to orchestrate widespread physiological changes via the anterior pituitary hormones. The CNS gets sensory input from all over internal and external environments.
And this input then directly influences those hypothalamic neurosecretory cells.
Right, which in turn regulate the endocrine cells of the pituitary and, through tropic hormones, many other glands. It's an elegant example of the brain integrating information to fine-tune the body's entire chemistry, allowing for precise adaptation to environmental cues.
And that fine-tuning extends to how hormones themselves modulate these control pathways. We talk about axes like the hypothalamus pituitary adrenal cortex, or HPA axis. Sounds like a chain of command.
It is, yeah. These axes illustrate the principles of hormonal and neural modulation really well. Negative feedback is probably the most widespread type of hormonal modulation.
That's the self-regulating thing.
Essentially, yes. High levels of a hormone in the blood will suppress its own secretion further up the control pathway. For instance, high levels of glucocorticoids, like cortisol from the adrenal cortex,
Will inhibit the release of CRH from the hypothalamus and ACTH from the anterior pituitary.
exactly, which helps stabilize hormone concentrations, keeps things from getting out of control.
But it's not always about turning things off. Sometimes hormones boost each other, right? Amplify the effect. Does that happen? And what's an example?
Absolutely. That's called synergism, when one hormone amplifies the effect of another. A good example involves ACTH secretion. Vasopressin, by itself, doesn't do much to ACTH-secreting cells in the anterior pituitary.
Okay.
But when it acts together with CRH, the main releasing hormone, the secretion of ACTH is much, much greater than it would be from CRH alone. They work better together than just the sum of their parts.
Interesting. So we've talked about hormones amplifying each other, but what if one hormone is absolutely useless without another? Does that happen? That kind of dependence?
Yes, that's called permissiveness, and it's critical in some cases.
Yeah?
Cortisol, a glucocorticoid, is a great example.
Yeah.
It doesn't directly cause vasoconstriction, but it's required for norepinephrine to cause strong vasoconstriction.
Which is essential for maintaining blood pressure, especially under stress.
Totally essential. Without even basal levels of cortisol, like in a condition called adrenal insufficiency, a person could be at severe risk if they experience a stressor like a hemorrhage that requires systemic vasoconstriction. Cortisol basically permits norepinephrine to do its job effectively.
Got it. Permits. Makes sense. And then there's the opposite. Antagonism. Hormones working against each other.
Right. That's when one hormone opposes the action of another. Insulin and glucagon are the classic example,
Insulin lowers blood glucose.
by promoting cellular uptake and storage. While glucagon raises blood glucose by stimulating its release from the liver, their balanced actions maintain stable blood glucose levels.
And other hormones can jump in too.
Oh yeah. Epinephrine, for instance, also raises blood glucose, acting antagonistically to insulin, and actually works synergistically with glucagon to amplify that effect. It's a remarkably complex but robust system with multiple checks and balances.
Beyond these hormonal interactions, neural input also modulates endocrine pathways. How does our brain directly influence these systems? We touched on the hypothalamus.
It's a very direct connection. Sensory input, like perceiving a threat which causes stress, directly influences those neurosecretory cells in the hypothalamus, like the ones that secrete CRH.
And what about internal clocks?
Yeah, biological clocks in the brain, like the ones driving our circadian rhythm, profoundly influence hormone secretion patterns. Cortisol levels, for instance, naturally peak in the early morning and are lowest in the evening, aligning with our wake-sleep cycle.
The ingenuity of this system is that hormones are often released in these intermittent bursts, right? Pulsatile secretion. Why is that important? Seems inefficient.
It seems counterintuitive, but it's thought to be a clever adaptive strategy. If hormones were just continuously present at high levels, target cells would typically downregulate their receptors. They become desensitized.
Ah, so the bursts keep the target cells listening.
Exactly. Pulsar release helps prevent this desensitization, maintaining the target cell's responsiveness over time. So the minute-to-minute blood levels of hormones are this complex dance between neural input, hormonal feedback, synergism, antagonism, all designed to keep the system responsive and stable.
Okay, shifting gears a bit. Let's delve into adaptation and survival, starting with the mammalian stress response. Sounds like a big one. A generalized physiological constellation for survival in threatening situations.
It is. It's an immediate, coordinated adaptation, preparing the body for what's often called fight or flight.
And stressors aren't just negative things like wounds or extreme temperatures.
Surprisingly, no. Even positive novel situations like exploring a new environment or even basic things like feeding and sexual activity can stimulate parts of the stress system. The body coordinates its response through both the sympathetic nervous system, the rapid response, and the HPA axis we just talked about, the slightly slower, more sustained response.
And there are distinct phases to this response. It's almost like our bodies are still preparing for a saber-toothed tiger, but instead it's just endless emails and traffic jams. How does that mismatch affect us in the long run?
That's a huge issue in modern life, absolutely. But looking at the evolved response, in the short term, those early effects kick in within about a minute,
Okay.
driven by catecholamines, epinephrine, and norepinephrine.
Adrenaline rush.
Basically, you
Yep.
see rapid increases in heart rate, breathing rate, blood pressure. Energy mobilization is huge glucose, and fatty acids flood the blood. Alertness and cognition sharpen. Blood gets diverted from the skin, and digestion gets suppressed. Priorities shift.
And the hormones coordinate this fuel supply.
Yeah, epinephrine and norepinephrine also cleverly inhibit insulin secretion and stimulate glucagon secretion. This ensures that glucose is plentifully available for the brain and muscles, which need it urgently. ACTH also gets released quickly and seems to facilitate learning during stressful events, and the co-secreted beta-endorphin provides some natural pain relief, analgesia.
Then about an hour later, the second effects emerge, primarily driven by glucocorticoids, like cortisol.
Right. These generally reinforce the early actions. They drive further metabolic changes like breaking down proteins, protein catabolism, and making new glucose in the liver, gluconeogenesis, all about ensuring sustained fuel availability for physical exertion and the brain.
And they help with blood pressure, too.
Yes, they amplify that permissive effect on vasoconstriction we mentioned, which is crucial for maintaining blood pressure, especially if there's blood loss. In case of hemorrhage, vasopressin and aldosterone are also triggered to conserve fluid volume and maintain blood pressure. It's a multi-pronged defense.
What's truly remarkable here, and maybe less intuitive, is the intricate interaction between the nervous, endocrine, and immune systems during stress. How do they communicate so effectively?
It's a true web. Yeah, we're learning more about this all the time. When immune cells detect pathogens like bacteria or viruses, they release signaling molecules called cytokines.
Okay, we mentioned those as local signals earlier.
Right. But some cytokines can also travel through the blood to the hypothalamus and directly stimulate CRH secretion.
So the immune system is directly telling the brain's stress center, hey, we've got an infection.
Essentially, yes. It directly links the immune system to the HPA stress response. This achieves two critical goals. First, it mobilizes energy reserves needed to fight the infection. Fighting infection takes a lot of energy.
Makes sense. And the second goal?
Second, the resulting glucocorticoids, at higher concentrations, actually help to mute inflammation. They prevent the immune system from overreacting and causing excessive tissue damage. It's an integrated survival strategy. Fight the bug, but don't burn the house down doing it.
This suggests a much deeper connection between mind and body than maybe we traditionally thought. This whole field of neuroimmunomodulation. What more might we discover here?
Oh, it's a really exciting field. It's ripe for future investigations into how these systems interact not just in stress, but in maintaining everyday health, homeostasis, and how disruptions might contribute to disease, and potentially how we could leverage these connections for new therapies.
But while the stress response is adaptive in the short term, it has significant downsides when it becomes chronic, as you alluded to with the modern stressors like traffic jams and emails.
Exactly. That's the key distinction. Short-term adaptive responses become maladaptive when the stressor doesn't go away.
So chronic stress sounds like a real problem then. What kind of problem?
It really is. Prolonged high blood pressure can lead to hypertension and cardiovascular disease. Continuous exposure to high glucocorticoids causes muscle wasting, thinning of bones, suppressed immunity making you more susceptible to infections, and even reproductive dysfunction.
And it affects the brain, too.
Critically, yes. Chronic stress can damage brain areas like the hippocampus, which
Oh.
is vital for learning and memory. That's a major concern.
And even in wild animals, glucocorticoid levels aren't just flat except when stressed. They fluctuate normally.
They do. They show fascinating seasonal variations, often peaking during high-energy periods like the breeding season or maybe just before winter. This highlights their role in priming the body for predictable seasonal challenges, whether it's high energy demands or anticipating specific events like disease outbreaks or severe weather. It's like a built-in seasonal physiological adjustment calendar.
Okay, let's transition now to how the endocrine system controls something fundamental. Nutrient metabolism in mammals. It's quite a challenge for the body to continuously provide nutrients despite only eating intermittently.
It truly is. Cells need fuel and building blocks all the time, and often in different proportions than what's in our last meal. Insulin is the major player here, the dominant hormone in the fed state, managing that short-term nutrient availability after eating.
Right. Stimulated by high blood glucose after a meal, also high amino acids, some gastrointestinal hormones chime in, and even parasympathetic nervous activity.
And it's known for its hypoglycemic effect, lowering blood glucose.
How does it do that, mainly?
It promotes the uptake of glucose, fatty acids and amino acids out of the blood and into storage tissues like muscle and adipose tissue, fat.
Using those GLUT4 transporters.
Exactly. Insulin triggers GLUT4 transporters to move to the cell membrane, allowing glucose uptake. This happens everywhere except for the brain, liver, and exercising muscle, which have different transporters that don't need insulin.
And inside the cells, insulin promotes storage.
Yes, it promotes the synthesis of glycogen, stored glucose, triglycerides, stored fat, and proteins while actively inhibiting their breakdown. Then, when insulin levels naturally decline between meals or during fasting, the body shifts gears towards mobilizing those stored nutrients. The dramatic effects of its absence are, of course, clearly seen in diabetes mellitus.
Then there's glucagon, insulin's counterpart, dominant in the unfed state or fasting.
Right. Glucagon is secreted by the alpha cells of the pancreas, mainly in response to low blood glucose. Also by sympathetic stimulation, and interestingly, high amino acid levels can also stimulate it.
High amino acids stimulate both insulin and glucagon. That seems weird.
It does seem weird at first glance.
Yeah.
We'll come back to that. Glucagon has a hyperglycemic effect. It increases blood glucose,
Ow.
primarily by stimulating glycogenolysis, breaking down stored glycogen in the liver, and gluconeogenesis, which is forming new glucose from non-carbohydrate sources like amino acids and glycerol, also mainly in the liver. It also promotes fat breakdown, providing alternative fuel.
Okay, back to the amino acid thing. What's the cleverness in how insulin and glucagon work together, especially when they sometimes rise simultaneously, like after a high-protein, low-carb meal?
Yeah, it seems counterintuitive, but it's actually a brilliant adaptation. Think about a meal that's mostly protein. It provides lots of amino acids, but very little glucose. Your muscles need those amino acids for protein synthesis, and insulin promotes that uptake and synthesis. But your brain strongly prefers glucose as fuel, and there wasn't much in the meal.
Ah, so the glucagon rise is needed.
Exactly. The simultaneous rise in glucagon ensures an output of glucose from the liver's glycogen stores, making sure the brain gets the glucose it needs, even while insulin is busy dealing with the amino acids. It's a subtle but crucial coordination to maintain fuel balance for all tissues.
That is clever. And other hormones play synergistic and permissive roles in metabolism too, right? It's not just insulin and glucagon.
Oh, definitely not. Growth hormone and glucocorticoids synergize with epinephrine to enhance lipid breakdown, especially during fasting or exercise. And those background levels of glucocorticoids are also permissively essential, as we said, for preventing blood glucose from plummeting during fasting or stress. They're required for glucagon and epinephrine to exert their full glucose-raising effects.
And others.
Thyroid hormones influence metabolic rate overall, and androgens, like testosterone, also promote protein synthesis and growth. It's a complex, multi-hormonal system designed for continuous nutrient supply, constantly adapting to feeding, fasting, exercise, stress, all sorts of conditions.
Okay, equally critical is maintaining salt and water balance. This is fundamental for blood pressure and just overall cellular function.
Absolutely critical. The body must tightly regulate the volume and the salt concentration, osmolarity, of its extracellular fluid. Antidiuretic hormones, like vasopressin or ADH, produced up in the hypothalamus and released from the posterior pituitary, are key players here.
Antidiuretic, so less peeing.
Basically, yes. Their primary role is to conserve water by limiting urine production in the kidneys.
How does vasopressin do that specifically?
It stimulates the insertion of special water channels, called aquaporin-2 or AQP2, into the cell membranes of certain kidney nephron cells, the collecting ducts mainly.
Like opening little doors for water.
Exactly. These channels allow water to be reabsorbed from the forming urine back into the body. This is a crucial mechanism for conserving water when, say, the extracellular fluid osmotic concentration is high, too salty, or when blood volume is low, like after a hemorrhage.
Okay. And if blood pressure drops significantly, another powerful system kicks in. The renin-angiotensin-aldosterone system, or RAS. That's a mouthful.
It is, RAS for short. This system is designed specifically to combat low blood pressure.
Which begs the question, how does the body specifically correct for low blood pressure using this complex RAS system? Walk us through it.
Okay. When low blood pressure is detected by specialized juxtaglomerular cells in the kidney, they release an enzyme called renin.
Step 1, renin.
Step 1. Renin then acts on a protein circulating in the blood called angiotensinogen, clipping off a piece to produce angiotensin I.
Step 2. Angiotensin I, still not active though.
Not really. Angiotensin I is then converted by another enzyme called ACE, angiotensin-converting enzyme, primarily in the lungs, into angiotensin II.
Step 3, angiotensin II. And this is the active player.
This is the powerhouse. Angiotensin II has multiple potent effects, all aimed at raising blood pressure and fluid volume.
Such as?
Okay, first, it stimulates the adrenal cortex, the outer part of the adrenal gland, to secrete aldosterone. Aldosterone is a steroid hormone that tells the kidneys to conserve sodium and excrete potassium. Water follows sodium, so this helps retain fluid.
Okay. Conserve salt and water via aldosterone. What else?
Angiotensin II is also a powerful vasoconstrictor. It tightens blood vessels directly, which increases blood pressure. Third, it stimulates vasopressin, ADH release from the posterior pituitary, further helping water retention. And fourth, it promotes thirst at the level of the brain.
Wow. So it tackles the problem from multiple angles. Salt retention, water retention, vasoconstriction, and making you drink more, all working together to restore blood pressure.
Exactly. It's a very effective system.
And the story behind the discovery of this system's therapeutic potential is remarkable, involving snake venom.
It really is an incredible example of how basic animal physiology research leads to major medical breakthroughs. The venom of the Brazilian pit viper, Bothrops jararaca, contains peptides that potently block ACE.
The enzyme that makes angiotensin II.
Right, so the venom causes a catastrophic drop in blood pressure in its victims. Scientists studied these venom peptides to understand exactly how ACE worked, its active site. And that knowledge allowed them to design the first synthetic ACE inhibitor drug, captopril.
Which is now widely used to treat hypertension, high blood pressure.
Exactly. From snake venom to a life-saving medication. A fantastic example of translational research.
And just so the body doesn't overshoot, there's a counter-regulatory hormone, too, to lower blood pressure.
Yes, there is. Atrial natriuretic peptide, or ANP.
Atrial, from the heart.
From the atria, the upper chambers of the heart.
Yeah.
ANP is secreted by heart muscle cells when they get stretched, which usually indicates high blood pressure or high blood volume.
So it does the opposite of RAS.
Pretty much. It's the body's natural way to reduce fluid volume and lower blood pressure. It promotes sodium and water excretion by the kidneys. It also inhibits the release of vasopressin, renin, and aldosterone. And it can increase the filtration rate in the kidneys. It's the balance against the RAAS system.
Okay, one more balancing act. Endocrine control of calcium metabolism in mammals. Why is calcium so tightly regulated?
It is absolutely critical, especially for proper nerve and muscle function. Too little calcium in the extracellular fluid can cause nerves and muscles to become hyperexcitable, leading to muscle spasms or twitches. Too much calcium can cause lethargy, muscle weakness, and other problems. So the levels need to be kept in a very narrow range.
And there are 3 main hormones involved here.
Primarily 3, yes. Parathyroid hormone, PTH, active vitamin D, and calcitonin.
Okay, PTH. Secreted by the parathyroid glands, those little glands near the thyroid.
That's them. PTH is secreted when extracellular calcium levels get low. Its job is to raise calcium levels.
How does it do that?
It acts on 3 main targets. First, bone. PTH stimulates bone resorption, breaking down bone tissue to release calcium and phosphate into the blood. Second, kidney. It stimulates calcium reabsorption in the kidney tubules, preventing calcium loss in urine while promoting phosphate excretion. And third, it stimulates the activation of vitamin D in the kidneys.
Ah, so it links to the next player, active vitamin D, which is actually a hormone.
Yes, active vitamin D, technically 1025 dihydroxyvitamin D3 or 1025 OH2D3, is a steroid hormone. It's formed initially in the skin through exposure to UV light, then processed in the liver, and finally activated in the kidney.
And that final activation step is stimulated by PTH and low calcium.
Correct. Active vitamin D's main job is to promote the absorption of dietary calcium from the intestine. That's its primary role in boosting blood calcium. It also works synergistically with PTH to resorb bone and helps reabsorb both calcium and phosphate in the kidney.
So PTH and active vitamin D work together to raise calcium. What about the third one, calcitonin?
Calcitonin is secreted by specific cells, parafollicular or C cells, in the thyroid gland, and is released when blood calcium levels get high. It does the opposite of PTH.
So it lowers calcium?
Primarily, yes. Calcitonin mainly acts to oppose bone resorption, thus decreasing the release of calcium and phosphate from bone into the blood. It might also facilitate their excretion in the kidney.
Is it as important as PTH and vitamin D in humans?
That's the nuance. While its effects are very strong in lab experiments and in other mammals especially, interestingly, in pregnant or lactating females, where it might protect the maternal skeleton from excessive calcium loss, its direct day-to-day role in regulating calcium in adult humans seems less critical than PTH and active vitamin D.
But it still has uses.
Oh, yes. Clinically, salmon calcitonin, which is potent in humans, is used as a drug to treat conditions like osteoporosis, Paget's disease of bone, and hypercalcemia, partly by inhibiting bone breakdown. Again, comparative physiology informing human medicine.
Okay, let's totally shift gears now, move into parallel worlds. We need to talk about invertebrates too. Insect metamorphosis is a fantastic case study showing that endocrine systems are just as vital and complex outside of vertebrates.
Absolutely. It's a compelling example of convergent evolution, completely separate evolutionary path arriving at sophisticated hormonal control systems. Insect transformations are just amazing.
You have the two main types, right?
Right,
Hemimetabolous.
hemimetabolous insects like cockroaches or grasshoppers, they go through gradual metamorphosis. Egg hatches into a nymph, which looks kind of like a small, wingless adult. It molts several times, getting bigger with each molt. Those stages are called instars. And then finally molts into the winged reproductive adult. Gradual change.
But then there's the truly dramatic stuff, holometabolous metamorphosis. Moths, butterflies, beetles, flies.
This is what most people think of when they hear metamorphosis. It's a complete transformation. Egg hatches into a larva like a caterpillar or maggot, which is often specialized for feeding and growth. It molts through several larval instars.
Often the stage that eats our crops or gets into our food.
Very often, yes. Then the larva transforms into a pupa. Think chrysalis or cocoon. Inside the pupa, there's a massive reorganization. Most larval tissues break down, and adult structures develop from little clusters of cells called imaginal discs.
Like a biological restart button.
Kind of. And finally, the adult insect emerges from the pupa, often looking totally different from the larva, specialized for reproduction and dispersal. The silkworm moth is a classic example study for this complete life cycle.
And this whole incredible process is controlled by a fascinating hormonal trio.
Yes, three key players orchestrate it. First, prothoracicotropic hormone, or PTTH. It's a neurohormone released from the brain.
PTTH from the brain. What does it do?
It acts like a trigger. It initiates the molting process by stimulating a pair of endocrine glands located in the thorax called the prothoracic glands. Its release is often influenced by environmental cues like day length and temperature, all integrated by the nervous system.
Okay, so PTTH tells the prothoracic glands to act. What do they release?
They release ecdysone. Ecdysone is a steroid hormone.
Ah, a steroid like cortisol or testosterone in us.
Chemically similar class, yes. Ecdysone itself isn't the most active form. It gets converted to the more potent 20-hydroxyecdysone, or 20E, right at the target tissues, mainly the epidermis, the outer cell layer.
And 20E is the signal to molt.
Yes. 20E stimulates the epidermal cells to secrete enzymes that digest the inner layers of the old cuticle, and then to synthesize the components of a new cuticle underneath. It sets the stage for shedding the old exoskeleton.
Okay, so PTTH starts it, 20E drives the molt. What about the third hormone, juvenile hormone, or JH? This one sounds intriguing. What's its role in this dramatic transformation?
JH is where the real magic of metamorphosis control lies. It's chemically different, a terpene, not a steroid or peptide, and it's secreted by another pair of endocrine glands near the brain called the corpora allata.
And its job is?
Here's the clever part. JH maintains juvenile characteristics. It essentially prevents metamorphosis from happening too early. The relative levels of JH and 20E determine the outcome of a molt triggered by 20E.
How so?
If JH levels are high when 20E peaks, the insect molts, but it molts into another juvenile form, like larva to bigger larva or nymph to bigger nymph. But if JH levels are low or absent when 20E peaks, then that molt results in metamorphosis either to the pupa, in holometabolous insects, or directly to the adult, in hemimetabolous insects, or the final pupa to adult molt.
So it's the decline in JH that permits metamorphosis to occur.
Exactly. It's the balance or ratio between 20E, the molt signal, and JH, the stay young signal, that dictates the developmental pathway.
And experimental methods really helped figure this out, didn't they?
Absolutely. Classic experiments showed this beautifully. If you surgically remove the corpora allata, the source of JH, from young larvae, they would undergo premature metamorphosis at the next molt, becoming tiny pupa or adults.
Wow. And the opposite.
Conversely, if you implanted active corpora allata or applied synthetic JH to a final instar larva, which normally has low JH, you could prevent metamorphosis and cause it to molt into an extra, often giant, larval instar.
That understanding must have real-world applications.
Huge ones. For instance, silk growers sometimes treat silkworm larvae with JH analogs, synthetic chemicals that mimic JH activity. This keeps the larvae feeding and growing longer, producing larger cocoons and thus more silk.
More silk, more money.
Right. And on the flip side, JH analogs like methoprene are used as insecticides. They mess up the hormonal balance. Applied to larval habitats, they prevent the insects from developing into reproductive adults, effectively controlling the pest population by disrupting their life cycle. It's a targeted approach.
A very clever pest control. Now, the actual shedding of the old cuticle, the ecotasis process itself, is also tightly orchestrated, right? Not just triggered by 20E.
It is. It requires another whole suite of hormones to coordinate the behaviors and physiological changes needed. Specific neurohormones trigger the release of ecdysis, triggering hormone, ETH, from specialized cells. ETH then acts on the nervous system to orchestrate the specific muscle contractions needed to break free from the old cuticle.
And then hardening the new one.
Yes, another hormone called bursicon plays a key role in the tanning, hardening, and darkening of the new cuticle after excitis, and also in expanding the wings in newly emerged adults. It's a precisely timed chemical and behavioral sequence.
And JH isn't just for juveniles, it comes back in adult.
It does. In many adult insects, JH secretion resumes, but now it often acts as a gonadotropin, a hormone that supports reproduction. It's needed for the production of fertile eggs in females and sometimes sperm in males, and it can also stimulate the production of pheromones used for mating. Its role shifts after metamorphosis.
And here's where it truly gets interesting, connecting back to other fields. Understanding insect life cycles, driven by these hormones, has practical applications you might not expect, like in forensics and even medicine.
It's remarkable, isn't it? Forensic entomology is a great example. Blowflies are often among the first insects to arrive on a dead body.
Because they lay eggs on decaying matter.
Exactly. Those eggs hatch into maggots, the larvae, which feed on the tissue. These maggots go through predictable larval instars, driven by ecdysone and JH, eventually pupating. By identifying the insect species present, determining their exact developmental stage, which instar, pupa, and factoring in environmental conditions like temperature.
Forensic scientists can work backward to estimate the postmortem interval, the time of death, often with surprising accuracy.
Precisely. It relies directly on understanding their hormonally controlled development rates.
And even more surprisingly, sterile blowfly maggots are used in modern medicine for cleaning wounds.
Yes. It sounds gruesome, but it's incredibly effective in certain situations. Maggot debridement therapy, or MDT. Specially reared sterile maggots are applied to non-healing wounds, like diabetic foot ulcers.
And they just eat the dead stuff.
They only consume necrotic or dead tissue. They secrete enzymes that liquefy it, and then they ingest it. They leave healthy tissue alone. Often, they provide superior cleaning compared to surgical instruments, especially in complex wounds.
As long as they're sterile.
Absolutely. They are specifically sterilized to ensure no pathogens are introduced. It's an incredible example of harnessing a natural biological process, deeply rooted in insect physiology and endocrinology, for a modern medical application, finding solutions in unexpected places.
Okay, wow. We've unpacked a vast amount of ground today. It really reveals the incredible complexity, but also the elegance of these endocrine and neuroendocrine systems across the whole animal kingdom.
It really does. And if you try to connect all these details to the bigger picture, you start seeing some recurring themes, some prevailing patterns that consistently emerge in animal physiology.
Like what? What are the big takeaways?
Well, first, I think it's clear that no single complex physiological system, metabolism, stress, reproduction, whatever, is controlled by just one hormone. It's always a symphony, a network of many interacting hormones.
Right. Multiple inputs, multiple controls.
Second, the flip side. A single hormone often affects multiple different systems or target tissues. It creates this ripple effect throughout the body. Think about cortisol affecting metabolism, immunity, brain function, blood pressure.
Very widespread effects. Okay, third pattern.
Third, hormones rarely act in isolation. They constantly interact with each other, sometimes synergistically, amplifying effects, sometimes permissively, where one is needed for another to work, and sometimes antagonistically, opposing each other to maintain balance.
That interaction piece is key.
Fourth, the endocrine system is inextricably linked with the nervous system. We saw that clearly with the hypothalamus and pituitary. And increasingly, we understand its deep connections with the immune system too, forming this true web of communication that maintains homeostasis and allows the animal to adapt to challenges.
Nervous, endocrine, immune, the big three communication networks all talking to each other. And finally,
many of these signaling molecules are incredibly versatile. A substance might serve as a hormone circulating in the blood in one context, but act as a neurotransmitter across a synapse, or even a local paracrine signal in another part of the body or at a different time. The same molecule can wear different hats.
So reflecting on all that, especially that intricate web of communication between the nervous, endocrine, and immune systems, it makes you wonder, doesn't it? What new mind-body interactions might future research into this field of neuroimmunomodulation reveal? Could it perhaps even unlock treatments for diseases we currently struggle with, like autoimmune disorders or chronic inflammatory conditions?
It's a profoundly important question. The potential seems enormous. And thinking back to the start, how might continued deep dives into the extreme physiologies of animals like that hibernating grizzly bear, or even the detailed hormonal control in insects? How might that continue to inspire completely novel approaches in human medicine, or help us understand adaptation itself in our rapidly changing world?
Lots to think about there. It really underscores the value of understanding these fundamental biological processes across all forms of life.
Indeed. Well, that brings us to the end of this deep dive. Thank you, as always, for being part of the Last Minute Lecture family.